Now that we've identified the proton-to-proton and proton-to-carbon correlation in the molecule, we can now therefore use HMBC to correlate protons with surrounding carbons. Now we have here the HMBC spectra of the molecule. Now the table on the right shows the summarized HMBC correlations on the structure. The protons 5, 8, 11, and 9, and with their corresponding carbon correlations verifies the presence of the aromatic ring that have a methoxy group. Also, proton 7' and 5' with their corresponding carbon correlations support the presence of an imidazole ring in the structure. Detailed analysis of the hydrogen-carbon correlation in HSQC and HMBC revealed that the compound is alkaloid in nature, containing histamine and vanillic moieties connected to a 2-methoxy-acrylamide bridge. A NOE study is also conducted on the molecule. In this experiment, the proton 10 with a selective pulse of 3.6 ppm was excited. So, as you can see here in the spectra, a strong negative peak is observed at the frequency of selective excitation. Now, positive peaks then rise due to the nuclear Oberhauser effect. This revealed the proximity of two aromatic protons with a chemical shift of 7.36 for proton 5 and 7.14 ppm for proton 9 to the methoxy group, which resonates at 3.6 ppm, confirming the Z configuration of the molecule across the olefinic double bond. The proposed structure was further verified by tandem MS-MS. Having an amide bonding structure, alpha cleavage occurred at different positions to produce ladder ions A, B, C, and XYZ. Accurate measurements of mass-to-charge ratio values of different fragments and corresponding neutral losses of CH3OH, H2O, CO, and CH3 further supported this proposed structure. By connecting all the puzzle pieces together collected from IHD, HPLC, mass spectrometry, and NMR, the structure was successfully elucidated, and its figure is shown here. Having a molecular weight of 318.1450 grams per mole, its IUPAC name is ZN21H imidazole 4-ethyl 3,4-hydroxy 3-methoxyphenyl-2-methoxyacrylamide. The results also collectively suggest a structure of the venom alkaloid, which is similar but not identical to the natural hypotensive product pyridohistamine that is found in Ephedephra roots. Having elucidated its structure, the venom alkaloid was then synthesized from vanillin and methyl 2-methoxyacetate via Claisen-Smith condensation using a strong-base lithium diisopropylamide (LDA) that was generated by reacting N-butyl lithium and diisopropylamine in situ. The LDA catalyzed the condensation reaction, which was then treated with benzene sulfonyl chloride to form the addition product. This intermediate was subjected to an in situ elimination reaction catalyzed by triethylamine base to yield an unsaturated Z-carboxylic ester, which was hydrolyzed to form the unsaturated carboxylic acid. And lastly, it was aminated by histamine, producing the alkaloid with a 73% yield. The synthesized alkaloid was characterized by NMR and mass spectrometry and found to be identical to the wild-type venom alkaloid. As one of the major components of the venom, the researchers assumed that this may be the alkaloid that the scorpion was using to paralyze its prey. The alkaloid was then subjected to biological assay. Pilot experiments were conducted by the researchers to test the hypothesis. The alkaloid was injected to several crickets with varying dosages, up to 50 micrograms. But surprisingly, none of the crickets have been paralyzed by the alkaloid. This result suggests that even though the alkaloid is abundant in the venom, it is not the component that the scorpion uses to paralyze its prey. Future researchers need to explore its biological role since it is still unknown. To know whether the alkaloid only exists on the telson of the scorpion or in any other region, researchers conduct a mass spectrometric study on the hemolymph that was extracted from the junction of the femur and patella. So this is the HRMS spectra of the hemolymph extracted. As you can see, no ion signal that corresponds to the identified venom alkaloid is observed. The weak ion signal at M/Z value of 318.1315 corresponds to a different chemical. This suggests that the alkaloid must be selectively produced in the telson of the scorpion. Also, The HPLC profile of the venom is maintained alive in the laboratory for 30 days, feeding on fricates is identical to that of the wild venom extracted just after collection from the field. These two results suggest that this alkaloid originates from the scorpion's metabolic process and not from directly what the scorpion ingests. To conclude this presentation, the alkaloid from the M. Gurtschy was successfully characterized and elucidated using different spectroscopic methods such as mass spectrometry and nuclear magnetic resonance. It is the first ever alkaloid to be reported from a scorpion. Interestingly, the alkaloid is composed of vanillin and histamine moieties, which are used in many pharmaceuticals. As the structure of the venom is very close to hypotensive agent ferulohistamine, it can be investigated further in the future to explore whether it has the same hypotensive property. Notably, the alkaloid from the scorpion was found to be non-toxic to mice and human, and its non-toxicity is very promising if used in pharmaceutical drug in the future. With this as a candidate for evaluating its medicinal effect, the researchers developed a cost and effective method for chemically synthesizing the alkaloid from vanillin and histamine precursors. And that ends our presentation. Thank you for listening.